Alright, welcome everybody. I wanted to make this video of some frequently missed questions. So first is the events of synaptic transmission. So this is very similar to what happened with the neuromuscular junction. The action potential reaches the synaptic knob, then these voltage gated calcium channels open, then calcium enters the synaptic knob. Calcium triggers exocytosis of the vesicles. The neurotransmitters diffuse across the synaptic cleft, and the neurotransmitters bind to postsynaptic receptors. Then for the events of an action potential, so this is very similar to what we saw in muscles, but with neurons, the resting membrane potential is around minus 70. Something is going to happen to get it towards threshold. These are graded potentials or excitatory postsynaptic potentials. When it reaches threshold, this is the point where the voltage gated sodium channels are going to open. When those sodium channels open, sodium enters. The positive ion entering makes the cell more positive. It goes way up here. Then those sodium channels are going to inactivate. Once those channels are inactivated, and then the voltage gated potassium channels open, they just take longer to open. So now potassium channels are open, potassium leaves the cell, and those positive ions leaving the cell makes the cell become negative. And those potassium channels take a while to close, so it goes even below the resting membrane potential into what's known as hyperpolarization. And then as those channels close, it gets back to the resting membrane potential. So depolarization occurs when the inside of the cell becomes more positive or less negative than the resting membrane potential. Hyperpolarization occurs when the inside of a cell becomes more negative than the resting membrane potential. So that's what we saw back here. Then the first thing that's generated are graded potentials. These are generated on the dendrites in the receptive segment of the neuron. And so you can see some chemically gated channels here. These are chemically gated sodium channels. And so to generate an excitatory postsynaptic potential, or an EPSP for short, we would open a chemically gated sodium channel. Sodium then would enter the cell because of diffusion. Positive ions going in makes the cell more positive. However, these EPSPs, they're a type of graded potential. They're not very big and they don't last very long, as opposed to action potentials, which are very big and can travel very far, all the way down an axon. Inhibitory postsynaptic potentials, or IPSPs, they can be caused by chemically gated potassium channels opening that would allow potassium to leave, and as those positive ions leave, the cell would become more negative. It could also be due to chloride channels opening that would allow negatively charged ions to enter the cell, which again would make the cell more negative. Again, it's very short-lived, doesn't travel very far. So which of the following would be likely to cause hyperpolarization of a neuron? So again, that's making it more negative. Opening of a voltage-gated potassium channel, which again would allow positive ions to leave, or opening of a chemically gated chloride channel, which would allow negative ions to enter. And then which of the following would likely to cause a depolarization of a neuron? Opening a chemically gated sodium channel that would allow positive ions to enter, so depolarization, the cells becoming more positive, or opening a voltage gated sodium channel that would also allow positive ions in and make the cell more positive. And several presynaptic cells will touch a postsynaptic neuron. And here you can see the green ones are representing EPSPs and the red ones are representing IPSPs. And so then in the cell body, it has to integrate these, sort of combine them all together and decide if it's got enough of a stimulus to reach threshold or not. So that will bring us now to action potentials, and we're going to watch this movie, which hopefully will help you visualize and understand action potentials. Okay, so the inside of the cell has lots of potassium, outside of the cell has lots of sodium. This is a chemically gated channel right here. Acetylcholine just bound to it. Open the channel. Now sodium is going to enter because of diffusion. Then some of the sodium goes over here. This is a voltage gated channel. The positive charge is going to open this voltage gated channel. More sodium enters and it diffuses down here to the next voltage gated channel and we'll open it. More sodium enters and this sodium moves down here and opens the next voltage gated sodium channel. More sodium enters and these behind it are becoming inactivated, and this will continue all the way down the cell till we reach the end. So let's look at the figures from our book for generating action potential. So at the resting membrane potential, all the voltage gated channels are closed. We're at -70. Sodium enters from adjacent region. Again, this is because of chemically gated sodium channels opening. And then once we reach the threshold, that's the point where the voltage gated sodium channels enter. Then we're going to have lots of sodium entering, and we can see the cell becomes more positive. But then very quickly those sodium channels close, they become inactive, but this process just continues down the axon, all the way down the axon. And then following depolarization, we have repolarization. So the potassium channels open more slowly. Once they're open, potassium can leave. Those positive ions leaving makes the cell more negative, as we can see here. And it takes those channels a while to close, so the cell becomes more negative than the resting membrane potential. And then eventually those channels close and we get back to the resting membrane potential. So that's the steps of an action potential. That will help you with one of the questions on your assignment today. And now let's watch this video where we've added, this time we've added potassium channels, so repolarization we'll see, and we've added a sodium potassium pump. Again, acetylcholine is going to bind to a chemically gated channel here. Open the sodium channel, sodium will enter, and then this sodium is going to bind to this voltage gated channel, not bind to it, just comes in contact with it. Its positive charge opens this voltage gated channel, more sodium enters, that sodium comes down here. Meanwhile, acetylcholine is diffusing away, and back here this potassium channel is going to open and potassium is going to leave, so we've got repolarization happening back here. Meanwhile, up here this sodium channel is opening, and this is really happening faster than we can see it, so I had to slow it down for this animation. This potassium channel is going to open and potassium leaves back here. Meanwhile, this sodium is going to open the next voltage gated sodium channel, so depolarization is happening here. Meanwhile, this repolarization wave sort of follows it as potassium leaves, and again this would continue. So now there's a sodium channel that we can't see open, and this potassium channel is open. And then we're going to have the sodium potassium pump taking the sodium, pumping it out, and the potassium back in to get us back to the resting conditions. So hopefully those animations help you. The other thing I want to point out to help you with this is go to the Connect site. And go to Anatomy and Physiology Revealed. Go to the Nervous module, select the animations, and these interactives are really good. So this, I really like this nerve impulse one, the action potential generation, the action potential propagation, those are both good. And this shows you what's going on at a synapse, that's good. These interactives are really good. So this is going to show you the resting membrane potential. I showed you this one in class on Wednesday. This one is going to show you graded potentials, and then these two show you action potentials, and then again this is uh, the events at the synapse. So please do those. That will help you understand these processes. Alright, couple more things. Acetylcholine. You need to know how neurotransmitters are removed from the synaptic cleft. So with acetylcholine here, there's an enzyme called acetylcholinesterase, which breaks it down into acetate and choline. Then the choline is taken back up into the cell that released it, and it's recycled. Um, some other neurotransmitters have reuptake proteins. So, for example, serotonin you've probably heard of. The most commonly prescribed drug in our country is Prozac, which is an SSRI, which stands for Selective Serotonin Reuptake Inhibitor. And so there's a protein right here, and it would be when the serotonin is released, it gets taken back up into this cell and then it can be recycled. So what that drug does is it blocks this protein. So instead of going back into the cell it released it, the serotonin hangs out in the synapse longer. And it sort of tricks your body into thinking that you've released more serotonin than you have, which makes you happier. So because we know about synapses, we are able to design drugs that treat things like depression. Last thing I want to talk about is neuronal pools. So there's 4 types of circuits you need to know. This is a converging circuit where you have several axons all converging onto one. So one might be bringing taste or smell information, one is sight information, one is sound, like you hear the grill sizzling, you see food, you smell food. And they all send an output to this one, which is going to make you salivate to get ready to eat. The next one is a diverging circuit, pretty much the opposite of the converging circuit, where one neuron is going to affect on these 5 different neurons, and it could be more than 5. So each one of these is going to stimulate, say, one motor unit, because that's where we would see these as more in the motor systems. So a diverging circuit is one going to many. Another type of circuit is a reverberating circuit. In this one you have this neuron sending an input to this neuron, which sends an input to this neuron, and then this neuron sends input to this neuron, but it also sends input to this neuron, and then this neuron is going to send input or output out here, but also back to this neuron, which feeds back to this neuron. This neuron also feeds to this neuron, which feeds back to these 2 at the start. And so it produces this cycle. These are good for any cyclical activities like breathing. It's a circuit like this that keeps us breathing. So like this goes out to our breathing muscle, the diaphragm, and this keeps it activated every few seconds. Then the last one we need to know is a parallel after discharge circuit. So in this one you have, starts with 1 giving it an input out to this one, and to this one, and to this one, and then you have basically 4 neurons or lines of neurons working in parallel here, and then they all converge back onto this one. These are used in higher order thinking. We think math especially uses these. So those are the type of circuits you need to know for today. Or neuronal pools, they're called. Alright, so what you're going to do today is go to homework, and you're going to do the nervous system questions part 3. Feel free to work with somebody else on the class from this, but I don't want any groups bigger than 4. There are I want to tell you how many questions there are. 13 questions, and it's from 12.8 to 12.11, so they're all in your book. You can email me if you have any questions, and don't forget to take the quiz. I'm going to send a quiz link with this. So good luck with that, and I'll see you next week.